Sí, yo me podré quitar la... O sea, parece bien que me quite la mascarilla para la presentación, para estar más cómodo. Nervioso. Voy a colocar el agua en algún... Sí.
Okay, welcome everybody. This is the defense of the thesis, doctoral thesis of Cristina Delgado Sallent, supervised by Dr. Maria Victoria Puig Velasco at the Hospital del Mar Medical Research Institute, IBIM, and the thesis will be evaluated by a committee formed by Patricia Robledo from IMIM too, Miguel Valencia from the University of Navarra, and Albert Comte myself from IDIVAPS. And without further ado, Cristina, you can start your thesis presentation. You have 45-50 minutes to present your work, and then after that we're going to make questions. So if you wait, we're going to sit in front to be perfect. There for you.
Yeah.
Okay.
Good morning, everyone, and welcome to my thesis defense. Today I'm going to present my thesis project, Neural Substrates of Psychotic-like States and Cognitive Impairment in a Mouse Model of Schizophrenia, and subsequent rescue by antipsychotic drugs. As you may know, schizophrenia is a really severe mental disorder with 3 different types of symptoms. First, the positive symptoms like hallucinations and delusions, which are the most commonly known. There's also negative symptoms like the social deficits or anhedonia. And finally, there's also the cognitive deficits, such as in executive functions or working memory. And this is currently treated by antipsychotic drugs. There's 2 types of antipsychotic drugs. First, the classical or typical antipsychotic drugs like haloperidol that mainly target dopamine D2 receptors and are effective treating positive symptoms. There's also the atypical antipsychotic drugs like risperidone or clozapine that besides targeting these dopamine receptors, they also target serotonin receptors, in which the serotonin 1A receptors and serotonin 2A receptors, among others. They are effective treating positive symptoms, but they can also ameliorate negative symptoms and cognitive deficits. And these symptoms of schizophrenia have been extensively associated with different structural and functional abnormalities. And one way of measuring these functional abnormalities is via an electroencephalogram, or EEG. That it's a device that allows to measure the electrical brain activity from outside the scalp. And one way to analyze this electrical brain activity is via neural oscillations. Neural oscillations are rhythmic change of voltage generated by a synchronous population of cells. This allows an efficient and precise communication across the brain, and they can be divided into different frequencies like delta, theta, beta, low gamma, and high gamma. And for instance, in schizophrenia, it has been seen that there's alterations in delta and gamma oscillations that could be associated with hallucinations or cognitive deficits. Another way of measuring this electrical activity is via event-related potentials, which are these small changes of voltage generated by the direct stimulus on the brain, like an auditory sound. And they can be divided into early or late components depending on the distance to the stimuli. First, we have the early components that are mainly processed in the sensory areas, like the auditory cortex, and they have been associated with auditory perception. They have been reported to be altered in schizophrenia patients and related with positive symptoms, like the severity of hallucinatory traits. There's also the late components that are processed in more downstream areas of the brain, like the prefrontal cortex, and they have been associated with auditory processing and attention. These components also have been seen to be altered in schizophrenia patients and associated with cognitive deficits. Another type of event-related potential is the auditory steady-state response that consists that sounds at a certain frequency generated a response at that frequency in the brain, and this allows the measuring of pyramidal interneuron synchronization. Also in schizophrenia patients, this is abnormal, and it has been related with this impairment in the synchronization of the brain. Furthermore, a circuit that has been reported to be impaired in schizophrenia patients and related to cognitive deficits is the prefrontal hippocampal circuit. And since we can not study this circuit in depth in patients because we cannot put electrodes inside patients' brains, we have to resort to animal models. There's different animal models of schizophrenia. What all of them have in common is the schizophrenia-like symptoms. For instance, the genetic models, which would mimic a mutation that is present in a certain percentage of the patients. There's also the neurodevelopmental models that try to mimic the disruption of normal developmental processes which may underlie the onset of the disease. And then there's also the pharmacological models that use pharmacological agents to alter circuits that have been seen to be altered in schizophrenia, like the dopaminergic or glutamatergic circuit. As I previously mentioned, in the mice brain, we can put electrodes inside and record the local field potential, which is the electrical activity from the neurons surrounding the electrodes. Here we can implant the electrodes in the prefrontal cortex and the hippocampus, and again, we can obtain neural oscillations that can be divided into different frequencies. And in this case, theta and gamma oscillations, and not only the power in the local areas, also the phase amplitude coupling of the phase of theta oscillations with the amplitude of gamma oscillations and the functional connectivity between them have been associated with cognitive functions. For instance, they have been linked to this theta-gamma functional connectivity with an spatial working memory task, which has been seen to be impaired in a genetic mouse model of schizophrenia, accompanied by this impairment of theta and gamma functional connectivity. In our case, what we have chosen as a model, it's an Mnda hypofunction model, in which these models are based in that an hypofunction of NMDA receptors in the brain produce alterations in the dopaminergic, GABAergic, serotonergic system, which may onset the schizophrenia symptoms. And this is all based in that an NMDA antagonist, like ketamine, phencyclidine, also called PCP, or MK801, produce schizophrenia-like symptoms in humans and rodents. For instance, an acute dose of these drugs produce a psychosis-like states that in rodents is characterized by hyperlocomotion, stereotypic behaviors, and ataxia. And this is accompanied by different cellular and molecular alterations like disbalanced dopamine, dysfunction of PB interneurons, and excitation of pyramidal neurons. And these alterations also have been associated with neurophysiological ones, like disrupted theta and gamma activity and increased high-frequency oscillations. High-frequency oscillations are oscillations that go from 100 Hz to 200 Hz, so it's fast oscillations. These symptoms can be treated by antipsychotic drugs. There's also the subchronic model that it has been reported that it can mimic negative symptoms, but also cognitive deficits, like impairment in recognition memory, working memory, reversal learning, cognitive flexibility, or sensory-motor learning. And this is also accompanied with cellular and molecular alterations that were similarly to the acute ones, but more permanent. Again, the imbalanced dopamine, reduction of PB interneurons, and disinhibition of pyramidal neurons. However, the neurophysiological alterations in this subchronic model have not been described, and is one part in which I want to focus my thesis, to understand which are the neurophysiological alterations and how these could be associated with these cognitive deficits. Furthermore, some of these cognitive deficits can be restored by atypical antipsychotic drugs. As previously discussed, atypical antipsychotic drugs target serotonin receptors. It produces agonism in serotonin 1A receptor and antagonism in serotonin 2A receptors. And these both receptors are strongly present in the prefrontal cortex and the hippocampus, where pyramidal cells can express both, and interneurons can express one or the other. Furthermore, in a previous study of the lab, in a normal mice brain, we have shown that 1A agonism by DPAD, 2A antagonism by M100 produce inhibitory effects while enhancing delta oscillations similarly to the atypical antipsychotic drugs risperidone. Alloperidol also produces similar effects with a lesser inhibition, since dopamine D2 receptors are not that much present in this circuit. Therefore, the main aim of my thesis would be to unravel the neural dynamics of prefrontal hippocampal circuits under psychomimetic conditions in the PCP mouse model of schizophrenia, and evaluate the effects of antipsychotic drugs. For doing that, we have divided in two specific aims, and the first one would be to investigate psychotic-like states induced by acute PCP administration and the rescuing abilities of antipsychotic drugs, which correspond with Chapter 1 of my thesis book. Okay, in order to do that, we have started with normal adult black 6 mice, in which we implanted 3 electrodes in the prefrontal cortex and 2 electrodes of the hippocampus. Once they have recovered for the surgery, we have recorded them in their home cage. And we have done 2 different protocols. The first one is to assess the psychotic-like states produced by PCP. This protocol consisted in 15 minutes baseline of the mice freely moving in his home cage, followed by a PCP injections and a recording for another hour. The color line represents the moments of quantifications and were used for the statistical analysis. Then there was the other protocol to assess the effects of antipsychotic drugs and serotonin components in the psychomimetic-like effects produced by PCP. There is a start by a 30-minute baseline, followed by an internal control of saline that was recorded for another 30 minutes. And after this hour, we injected PCP, waited for 15 minutes, and injected one of the atypical antipsychotic drugs, risperidone or clozapine, the classical or typical antipsychotic drug, alloperidol, the serotonin 2A antagonist, M100, or the serotonin 1A agonist, DPAD. Again, the color lines represent the moments of quantifications, and they were used for the statistical analysis. Once all this data was recorded, we start with the analysis of it. And we analyze the local field potentials and the spiking activity. In the local field potentials, we divided them in different frequency bands: delta, fast delta, slow theta, theta, beta, low gamma, and high frequency oscillations. And the first part, we analyze the local activity. One way of analyzing the local activity is via power spectral analysis that measures the amplitude of these frequency bands and oscillations. This could be represented in this plot, like as you see here, which is an spectrogram in which the x-axis represents the time, the y-axis represents the frequency, and the color represents the intensity of the power. They could also have these lines on the top that are the accelerometer, which is the movement of the mice. Another analysis that we do is the firing rate that could be done by multi-unit analysis, which is the activity of all the neurons surrounding the electrodes, and single unit analysis, which is the activity of the isolated neurons from the electrodes. And to see the phase locking between this firing rate and the local field potential, we use a pairwise phase consistency measures that will allow us to see this phase locking. We next analyze the connectivity between the prefrontal cortex and the hippocampus, and we use different analyses, like the phase amplitude coupling that measures the modulation index, which can be measured locally or cross-regionally. And this measures how the phase of slow frequencies can modulate the amplitude of fast frequencies. Another measure of connectivity is the weighted phase lag index, which is a non-directed measure of phase synchrony. This could also be plotted by this color plot similar to the spectrograms or different types of quantifications. And the last connectivity measure that we use is the phase slope index, which is a directed measure of phase synchrony, in which a prefrontal to hippocampal directionality will be represented in green or as positive values, whereas an hippocampal to prefrontal directionality will be represented in red or negative values. So the first thing that we wanted to check in our results is if after the implantation and the surgery, we still have these psychotic-like states produced by PCP and the behavioral effects, and how this was affected by risperidone. So as observed here, PCP produced hyperlocomotion, increase of stereotypic behaviors, and ataxia that is represented by partial reductions of the accelerometer rates, while risperidone produced these immobility states in which the hyperlocomotion is reduced, also the stereotypic behaviors, but not the ataxia, and it's represented by this complete reduction of the accelerometer rates. Once this was established, I'm first going to describe the circuit under baseline or saline conditions. First, I would like to measure that these kind of spectrograms are CIRA score spectrograms. That means that they have been normalized from the baseline. So here we observe the changes from respect of the baseline, an increase or a decrease. So as it was previously described, we see these hippocampal theta oscillations that have been strongly associated with locomotion during baseline and saline conditions. There's also this, in the hippocampus, this theta, beta, gamma phase, spike LFP phase locking that is present under baseline conditions. We next investigated in the phase amplitude coupling, where we observed this theta gamma coupling in the hippocampus and in the circuit that was present through baseline again and saline conditions. And we next assessed the effects on connectivity, whereas we see this theta phase synchrony, as in here, that goes from the hippocampus to the prefrontal cortex. So once we established this baseline circuit, we wanted to assess how PCP could disrupt this circuit, producing these psychotic-like states. So as observed here, already with PCP in the raw local field potentials, we can see changes on the activity pattern in the prefrontal cortex and the hippocampus. We next assess the results in the power. As observed here, PCP produced a boost of prefrontal activity at delta, gamma, and high-frequency oscillations, whereas it completely disrupts the theta and middle range oscillations in the hippocampus. We next assess the spiking activity, in which we observe that PCP doesn't produce any changes in the firing rate. However, it produces alterations in the spike LFP coupling, enhancing delta and gamma oscillations in the prefrontal cortex and desynchronizing the theta, beta, gamma phase locking in the hippocampus. We next investigated the effects on the phase amplitude coupling, And as seen here, PCP again produced a boost of delta high-frequency oscillations coupling in the prefrontal cortex and the circuit, while completely desynchronizing the theta-gamma coupling in the hippocampus. We next investigated the phase synchrony, and as observed here, there's these theta oscillations that were completely desynchronized, and starts appearing this delta synchronization that is accompanied by a gamma high-frequency oscillations hypersynchronization, whereas there's a disruption of the beta low gamma synchrony. And then this is related with the directionality results, where we observe that this hippocampal to prefrontal directionality at theta range is disrupted, enhancing this delta prefrontal to hippocampal directionality. So as a summary of this, PCP produced a boost of prefrontal activity, desynchronized the hippocampus, and disrupted prefrontal hippocampal circuits. Once we establish how are the effects produced by PCP, we next assess how they can be affected by antipsychotic drugs. And we first investigated the results in the power, whereas both atypical antipsychotic drugs, risperidone and clozapine, could reduce these enhanced gamma and high-frequency oscillations in the prefrontal cortex while lowering delta oscillations, whereas alloperidol can. And none of these compounds can recover the disrupted or decreased activity in the hippocampus. We next assess the firing rate and spike LFP coupling, and as observed here, risperidone produced reductions on the firing rate in the prefrontal cortex and the hippocampus. And again, both atypical antipsychotic drugs can reduce the enhanced delta and gamma phase locking in the prefrontal cortex, whereas none of them can recover the disrupted theta, beta, gamma phase locking in the hippocampus. We next investigated the results on the phase amplitude coupling, whereas only risperidone can reduce the enhanced delta high-frequency coupling in the prefrontal cortex and the circuit, whereas clozapine and alloperidol can, and none of them can restore the normal theta-gamma coupling in the hippocampus and the circuit. And we next assess the effects on connectivity, whereas similarly we observe that atypical antipsychotic drugs can reduce the enhanced gamma high-frequency oscillations hypersynchronization, whereas none of them can recover the theta low gamma phase synchrony, and only clozapine is able to recover this theta prefrontal to hippocampal directionality and stop this delta prefrontal to hippocampal. So, as a summary of all these results, PCP produced a boost and hypersynchronization of prefrontal activity, desynchronized the hippocampus, and disrupted prefrontal hippocampal circuits. Atypical antipsychotic drugs can restore the hypersynchronization in the prefrontal cortex and the circuit, but they cannot restore the synchronization in the hippocampus or the circuit. And then the classical antipsychotic drug alloperidol had minimal effects in this circuit. This could be because their antipsychotic actions may be produced in other circuits. So therefore, we hypothesize that maybe it's the serotonin component of these atypical antipsychotic drugs what is making these strong effects in the prefrontal hippocampal circuit. So that's why we analyze how the serotonin 2A antagonist, M100, or the serotonin 1A agonist, DPAD, could affect the psychotic-like states produced by PCP. So as observed here in the power results, both components, similarly to risperidone and clozapine, can reduce these high-frequency oscillations and gamma synchrony, whereas slower in delta, and none of these components can restore the normal hippocampal activity. We next investigated the spiking activity, in which in both areas the spiking activity was reduced by both components, and again, similarly to atypical antipsychotic drugs, both components can restore the enhanced delta and gamma phase locking in the circuit, whereas they cannot restore the normal theta gamma phase locking in the hippocampus. We next investigated the effects on the phase amplitude coupling, whereas similarly to risperidone, both serotonin components can decrease the enhanced delta high-frequency oscillations in the prefrontal cortex and the circuit, whereas they cannot restore the normal theta-gamma coupling in the hippocampus. And where we see more difference between these serotoninergic components and the atypical antipsychotic drugs is in the connectivity, where both components have minimal effects in the hypersynchronization on the circuit and even disrupt more the directionality of the circuit. This may indicate that these serotonin components have a major role in typical antipsychotic drug activity, especially the local activity, whereas for the connectivity measures and other rescues, other receptors or the combination of them could be acting in the atypical antipsychotic drug actions. And these results have been recently published in this paper in Cerebral Cortex, so if you would like to check them with more detail. Now I'm going to start with the second part of my thesis, the second specific aim, which would be to investigate the neural correlates of the cognitive impairment induced by subchronic PCP and subsequent rescue abilities of chronic risperidone, which corresponded to chapter 2 and 3 of my thesis book. We again started for this subobjective by normal black 6 mice, in which we performed the same surgery that I explained before. The mice recovered for a week, and then we did baseline characterizations with a battery of tests: an open field exploration, the novel object recognition task to assess working memory and long-term memory, and 3 auditory tasks: the auditory evoked potential to study auditory perception and processing, the auditory steady-state response to study the synchrony of the circuit, and an oddball paradigm to assess the mismatch negativity. The mismatch negativity, it's a measure of attention that also has been reported that it's impaired in schizophrenia patients. Once the mice finished this characterization, they underwent to a subchronic PCP or saline treatment for 10 non-consecutive days, and we repeated this full characterization. Once the mice finished this characterization, they underwent to a risperidone or saline treatment for 14 days, and we did the whole characterization within 48 hours of finishing this risperidone saline treatment. Sorry about that. Okay. The task that we have used to assess working memory and long-term memory is the novel object recognition test. It's a test based in mice's innate preference for novelty. So if a mouse remembers a familiar object, it will explore more the novel object than the familiar object. In this case, it consists in 4 phases of 10 minutes each: an habituation of the mice to the maze, a familiarization in which we place 2 identical objects in each arm of the maze, and these objects start being novel, and the mice explore them, acquires the memory, and become familiar. And then we did a 3-minute test after the familiarization and a 24 hours after the familiarization test to assess working memory and long-term memory, respectively, in which we change one of the familiar objects by a novel object randomly. And again, if the mice remembered the familiar object, they explored more the novel object. This was synchronized with the electrophysiological data using a custom-made joystick, and the cognitive readout of this task is the discrimination index. The discrimination index is a ratio that measures the time exploring the novel object, then subtracts the time exploring the familiar object, and divided by the total amount of time. A good discrimination index was considered above 0.2, which represents 20% more of the time exploring the novel object than the familiar object. Below that, animals explore both objects similar amount of time, so it's considered a poor recognition memory. So now I'm going to describe the effects on behavior in both tests. First, in the working memory test, in which we observe, as previously described, that subchronic risperidone produces an impairment in this working memory where saline doesn't, and that a chronic risperidone treatment could recover this working memory impairment, whereas a saline treatment doesn't. And that this discrimination index is dependent on the number of explorations. In baseline, saline, and risperidone, mice explore more times the novel object than the familiar object, whereas in the subchronic PCP-treated mice explore both objects similar amounts of time, in which the number of familiar object explorations increase, being similarly to the novel objects explorations, indicating that the mice is not recognizing the familiar object and explore it as it was novel. We also observe similar results in the long-term memory test, in which, again, subchronic PCP produces a disruption of long-term memory that is not produced by saline, and chronic risperidone can recover, whereas saline can. And this is, again, dependent on the number of explorations in a similar way as I described in the working memory test. So once we have detected how PCP disrupted the memory and that chronic risperidone could recover, the next step would be to assess which neurophysiological biomarkers in the prefrontal hippocampal circuit are present in these subchronic PCP mice and how they are changed by risperidone. So in order to compare between baseline and the following state subchronic PCP in the same mice or the different group of saline, since subchronic PCP can induce hyperlocomotion, we try to compare the same brain state. So we use a resting state, which is quiet wakefulness, in which the mouse is in the open field, but awake and not moving. So in this case, what we observe here is that there's increased gamma in the prefrontal cortex, whereas gamma is decreased in the hippocampus, and this was accompanied by a decreased theta-gamma coupling in the hippocampus and also the circuit, whereas saline mice doesn't present these changes. We next assess if some of these neurophysiological biomarkers could be related with the behavioral response and the recognition memory deficits, correlating them with the discrimination index of the working memory and the long-term memory test. And what we observe here is that, in this case, prefrontal theta power only in the subchronic PCP-treated mice and not previously in their baseline conditions correlate negative with the discrimination index of the long-term memory test. And this is interesting because in a previous study of the lab with another cognitive impairment model, this correlation was already established, indicating that maybe there's a pathological theta oscillation that may be involved in the poor long-term memory. So once we established these biomarkers after PCP, we wanted to see how they were affected by the chronic risperidone treatment. And what we observe here is whereas chronic risperidone can partially rescue gamma oscillations and completely rescue this pathological theta correlation with the discrimination index during the long-term memory test. Whereas risperidone cannot rescue the decreased gamma oscillations in the hippocampus, neither the decreased theta-gamma coupling in the hippocampus or the circuit. So we next wanted to see if these results and neural substrates were only in the resting states or they were also present in more cognitive demanding states, like the explorations of the novel object recognition test. And what we observed is that the hippocampal decrease of gamma oscillations and the theta-gamma coupling in the hippocampus and the circuit were decreased across all explorations during the novel object recognition test, indicating that this is non-task specific and it's a permanent state. And again, this was not recovered by the chronic risperidone treatment. And these measures also correlate between them, indicate that maybe the same mechanism is generating these both deficits. Once we establish this brain state, the next step would be to understand how PCP disrupted the encoding of the memory in the working memory and the long-term memory, and how chronic risperidone could rescue these. But in order to start with this, the first step would be to characterize which are the neurophysiological biomarkers during baseline conditions in each phase of the NOR. So we are going to start describing the familiarization in which we compare the early explorations, the first 5 seconds, with the late explorations, the last 5 seconds. This would involve the novelty of the object, and here is with when the memory it's being acquired. So as observed here, there's a progressive increase of theta oscillations over the number of explorations. We also observe this difference in theta directionality, where early explorations are shown by a prefrontal to hippocampal directionality, whereas late explorations are shown by an hippocampal to prefrontal directionality. Then we assess how PCP changed this, and we observe that PCP increased theta oscillations over the whole time of explorations, not allowing this progressive increase, and it also disrupts this directionality of the circuit. We next assess how risperidone affects this circuit, and what we observe is that risperidone can recover this difference in directionality between early and late explorations while not recovering the enhanced prefrontal cortex theta power, indicating that risperidone could recover this biomarker, and having a better memory acquisition, this could lead to the better working memory and long-term memory performance. We next assess these biomarkers during the working memory test, in which we compare the first 5 seconds explorations with a novel object with the first 5 seconds explorations with the familiar object. And as we see these two directionality biomarkers, where again we observe this prefrontal to hippocampal directionality during novelty at theta range. And in this case, we observe this prefrontal to hippocampal high gamma directionality on the familiar object explorations, and the difference between novel and familiar object explorations that correlates with a better working memory performance. This high gamma directionality is disrupted by PCP, especially the familiar object directionality and also the correlations. And in this case, this wasn't recovered by risperidone, indicating that maybe other mechanisms or compensatory mechanisms or the previously good memory acquisition would be enough to recover the working memory performance. We next assess the effects on the long-term memory test. Again, we compare first 5 seconds of novel explorations with first 5 seconds of familiar explorations. We again observe this theta difference in directionality, whereas prefrontal to hippocampal directionality is also present during novelty. And in this case, there's also a hippocampal to prefrontal directionality at theta range, and also the difference between them that correlated with a better long-term memory performance. Another biomarker was found, which was based in low gamma, the low gamma synchrony, and again, the low gamma directionality, in this case only in the familiar object, hippocampal to prefrontal, that also correlated with a better long-term memory performance. Subchronic PCP disrupted these, especially the low gamma biomarkers and also the correlations, whereas it only partially disrupted the theta biomarkers. We next assess the effects of risperidone, and in this case, risperidone seems to use the low gamma phase synchrony and especially the difference between familiar and novel object phase synchrony that also correlate with a better long-term memory performance to encode this task. This plus maybe other mechanisms involving other circuits or compensatory mechanisms coming from the memory acquisition. So in order to summarize the changes that I just described, subchronic PCP during resting states produce enhancement on gamma oscillations in the prefrontal cortex, also disrupted theta, whereas in the hippocampus produce a decrease of gamma oscillations and a disruption of theta-gamma coupling in the hippocampus and the circuit. And this hippocampal state is non-target specific and present during all brain states. We also describe different biomarkers based on theta and gamma functional connectivity for memory acquisition, working memory, and long-term memory, in which novelty seems to be related with a prefrontal to hippocampal theta directionality, whereas again, prefrontal to hippocampal theta directionality at high gamma range could help to the working memory or short-term memory storage. And in this case, hippocampal to prefrontal directionality at theta and gamma oscillations help for the memory acquisition and long-term memory retrieval, respectively. Subchronic PCP disrupted most of these biomarkers, especially the ones related with memory acquisition and working memory, the ones more related with the familiar object explorations. Chronic risperidone can recover some biomarkers while not recovering the complete activity of the circuit, in which we observe the recovery of prefrontal cortex during resting states, also the memory acquisition biomarkers, and it may use other compensatory mechanisms to restore this working memory and long-term memory performance. As discussed previously, we have said that the schizophrenia patients present disruptions on auditory perception, processing, synchrony, and attention. And we wanted to study that in the subchronic PCP mouse model and how it's affected by risperidone. So we recorded the mice in their home cage in a soundproof box with a speaker inside that produces the sounds. In rodents, we can also observe these auditory evoked potentials, but at a faster time scales. We used 3 different protocols: the auditory evoked potential to assess auditory perception and processing, which consisted in clicks of sounds that produce these auditory evoked potentials. We also used the auditory steady state response to assess the synchrony of the brain, and this is generated by these 40 Hz blocks of sounds that produce this enhancement at power at that frequency that is consistent over trial, which will be seen in the intertrial coherence. And finally, we also did an oddball paradigm to assess mismatch negativity, which is the measure of attention. This paradigm consists in a set of standard tones at a certain frequency that were interrupted by a target tone and another frequency with a 25% rate. So these target tones will elicit a bigger auditory evoked potential response with respect of the previous standard tone, and the subtraction of this target tone from the standard tone will generate a peak here that is the mismatch negativity and this measure of attention. In this case, we are going to use the recordings done in the prefrontal cortex, that it's an area related with the auditory processing. So we first investigated the auditory evoked potential, and the first thing that we wanted to know if the subchronic PCP mice responded with auditory evoked potentials to the auditory tones the same times of baseline and salient. So here we calculated how many times they responded with this auditory evoked potential to the tones, and it's called the response ratio. So in this case, subchronic PCP reduced this response ratio compared to baseline and saline, indicating an impairment on this auditory perception. And also, the late components of the auditory evoked potential were disrupted, and this was accompanied by a reduction of the evoked firing rate in the subchronic PCP mice. We next investigated how this was affected by risperidone, and risperidone could partially recover this response rate and these late components of the auditory evoked potentials, indicating that PCP disrupt auditory perception and processing, and risperidone can partially recover that, as was previously seen in schizophrenia patients. We next assessed the effects on the auditory steady state response, in which, again, these 40 Hz sounds produce this increase in power that is consistent over trials, seen here in the intertrial coherence. And in this case, we don't observe a reduction with the subchronic PCP treatment, indicating that there would be no impairment in the synchrony. However, this is recorded in the prefrontal cortex, and the auditory cortex is the area where these responses were more present, and the number of mice that have done these auditory tasks were slower. So further studies with a larger number of animals and recording in the auditory cortex have to be done in order to confirm these results. And finally, the results on the mismatch negativity. As observed here, baseline and saline conditions produce this peak that is related with attention and the mismatch negativity, whereas it's completely disrupted in the subchronic PCP mice, indicating an impairment in attention, and this was not recovered by the chronic risperidone treatment, as it was previously seen in schizophrenia patients. So as a summary of these results, subchronic PCP produced alterations in the late components of the auditory evoked potential and mismatch negativity that could be related with impairments in auditory perception, processing, and attention, and only, like risperidone, only could recover partially the late components of the auditory evoked potential, as indicating only recovering of this auditory processing. However, this, as it was previously seen in schizophrenia patients. So finally, I'm going to read the conclusions if the committee will allow. From the part 1, psychosis-like state induced by acute phencyclidine correlated with hypersynchronization of the prefrontal cortex, desynchronization of the hippocampus, and disruption of connectivity of prefrontal hippocampal circuits. Atypical antipsychotic drugs, but not alloperidol, reduce prefrontal and prefrontal hippocampal hypersynchronization induced by phencyclidine. None of the substance restore hippocampal and circuit desynchronization. Atypical antipsychotic drugs' greater affinity for serotonin 1A and 2A receptors may explain their differential effects with alloperidol. And now from the second part, the encoding of novelty, memory acquisition, working memory, and memory retrieval in the novel object recognition task depend on theta and gamma functional connectivity within prefrontal hippocampal circuits. Subchronic phencyclidine produced recognition memory impairment that was associated with disruptions in both brain state and task-dependent theta and gamma synchronization and connectivity. Subchronic phencyclidine also disrupted auditory perception and attention, as observed in patients with schizophrenia. Chronic risperidone rescued memory impairment and auditory perception, but not auditory attention, as observed in schizophrenia patients. Selective correction of key pathological connectivity and evoked responses within prefrontal hippocampal circuits likely contributed to this rescue. And I finally wanted to thank all the people that have helped me during this process, and especially the people from my lab and my PhD supervisor, Dr. Vicky Puig.
Thank you for the presentation, and we're going to proceed now with a session of questions and answers. And I give the word to Patricia Robledo for the first round, if you don't mind.
Thank you. Hi, Cristina.
Hi.
Congratulations for this very nice talk. I really enjoyed it. And I also wanted to congratulate you and your tutor and all the team, of course, for a very good work, and especially combining these two techniques, very complex techniques, the electrophysiology and the behavior was really nice, very interesting work. So congratulations.
Thank you.
So I'm going to start asking you a little bit of questions and maybe commenting also on your thesis, the book.
Yeah.
Some general questions, and maybe I'll go later into more details.
Okay.
So the first thing that I kind of surprised me was the way you explained the hypothesis and the objectives. because it seemed like you divided the work in projects, and they seem like different projects, instead of being like a continuum. And I was wondering, why you did this. It kind of surprised me, because actually, in the way, in the significance that you wrote of the work, it seemed very clear to me that what you wanted to do was just, see what evaluate the neural correlates of two phenotypes that are associated with schizophrenia, which are, psychosis and memory deficits, and then the next objective was actually what the antipsychotics were doing in these two. So, in other words, the way that the objectives and it's just really kind of repetitive, So I was wondering if you could kind of comment on what your idea was in doing this, because it kind of dichotomizes the work, and and I was wondering if you just don't see it as a continuum, of these symptoms in schizophrenia. How do you view that, and why did you choose to do that?
Yeah, in that sense, like, I thought that I would wanted to, even if there's two different symptoms, I wanted to separate the psychotic part from more the cognitive part, since it got really complex, from the psychotic, the acute, and all the antipsychotics, compared with the more cognitive processes, the auditory, perception, and I thought that it would be easier to divide it in these two parts, to make it divided through symptoms, so positive cognitive symptoms that go through schizophrenia and antipsychotics, since in the first part we use more types of antipsychotics, serotonin components, and less behavior. It's more a description of the circuit, and in the second part we kind of involve more the cognition part, the behavior, and I thought it would be easier by dividing it that way.
Yeah, because it seems like you actually made a lot of emphasis on the PCP, the responses to PCP, and in that sense, can you just kind of why did you use PCP as a model of schizophrenia instead of other models, for example, that you explained as, for example, the neurodevelopmental or the genetic models? So what I'm asking is really what the relevance of PCP for schizophrenia compared to the other models that are available.
Yeah, and in this sense, I would like PCP in this sense. It's a more like a general model that we know that impairs a circuit that is impaired in schizophrenia and follows the glutamatergic hypothesis. So it's more straightforward in that sense, that the neurodevelopmental or genetic models that they are, I think all the models at the end are complementary and are needed, because the PCP could be an easier way to start describing the circuit, because we are sure that impair the circuit, that these are more schizophrenia-related phenotypes, whereas in the genetic model, I think we only are including a more percentage of the patients, that more, I think the genetic model that englobes more of the patients is at 4%. This is pretty low. And in the neurodevelopmental models, there's this variability that in which they could develop, you are just exposing them to risk factors of schizophrenia, like prenatal stress or social isolation. So in this sense, I feel it would be more variability in the circuit. So I think the first step would be to go from something that is disrupt the circuit. We know that there's these symptoms, and from that move to understand how other models would also similarly affect the circuit, and
Yeah,
That's
definitely. Yeah, there is a controversy also, but between PCP only produces cognitive deficits and not negative deficits, or I don't know. So, do you think it's a real good model of schizophrenia, being that it doesn't kind of reproduce the whole spectrum of symptoms in schizophrenia?
Yeah, that's like a really good question, and like this is like a currently ongoing topic. There's not the perfect model of
Right.
schizophrenia. There's like, I feel like none of the models could mimic such a complex disease.
Exactly.
So the disease at the end is really complex, and it's at the end it's these symptoms that I described before. If you feel some of these symptoms, you are categorized as a schizophrenic. But it doesn't mean like all schizophrenic people have the same defects, and what is the difference again of some defects from bipolar.
Right.
So I think these neuropsychiatric disease models are really difficult like to completely mimic the disease. We have, in the sense that we see the subchronic PCP, cognitive impairment, so like in that sense, let's focus on the cognitive impairment.
You can really, yeah.
And see what happens there, because like I feel like try to model the entire in mice especially, it would be like really complex.
Okay. Yes, I agree. I'll ask you some other questions at the end about the difference between acute and subchronic. But now I wanted to go into a little bit into the introduction, because I as a expert in electrophysiology, I did find that your introduction was lacking a little bit of visual effects or sort of thing, which is really interesting, because your presentation had really nice summary slides on the different electrophysiological biomarkers in that you were studying, and I was kind of missed that. It was very arid, the introduction, and in that sense, I was and you did have, for example, the ERP. The only figure in the introduction was the ERPs, the event-related potentials, and I thought it was out of place, because it just came out of nowhere. So I think that it would have been nicer to have some visual effects in the introduction. And in that sense, I was thinking about all these different biomarkers that you explained. In humans, for example, I'd like to seen what I saw in your presentation, which is thank you for that, for putting that in there. And I was wondering if, because you said that you could measure all these biomarkers in humans by electroencephalography, EEG, but there's also a lot of connectivity data for humans. And I was wondering how you do that. Is it with another paradigm, or is it with that second paradigm that you've mentioned, or is it that for example, do you get data from magnetic resonance studies, and kind of can you associate these two type of results? I was just out of curiosity.
Like in this sense, I'm like not an expert in the human research.
Oh,
so I don't completely understand it or know everything. And like in humans, you can record from or electroencephalogram from outside of the cells. There's also fMRI and other measures that can be done. Like what it doesn't allow you in humans is have the precise location that you have in mice.
Okay.
And there's also connectivity measures, but goes maybe from frontal to parietal, but
Not
it's
deeper.
not
Deeper
as
structures.
specific as it would be. And then you have the fMRI that it could be more specific,
Right.
but then you can not see that much the electrical activity. It's more visual and less temporal resolution.
Okay, good. So, okay, now I wanted to ask you next, going to a little bit of methods and more kind of specific questions. And I really had a trouble with your design and protocol design and your statistics. I had problems understanding and following. It was really difficult for me because again, we had a very few visual aids in the book. And so, you have on page 79, you have, a summary of a visual summary of your protocols, but A is the protocol, the acute administration of PCP, looking at the psychotic, pro-psychotic responses, and then the reversal by different types of antipsychotics, which is, I suppose there you had, I was wondering how many groups did you have here? I know that you did the antipsychotics, you did it within subjects. So every animal received all of the antipsychotics?
No. Every animal received...
Because you talked about a washout of a week.
Yeah, no, there's like, in that sense, maybe, sorry that it wasn't like that well explained in the book, that animals as much receive 3 different drugs. So, and they do these 3 different drugs. There's like many animals that just did like 2 or 3, like PCP risperidone, PCP alloperidol, PCP saline. So at the end there's a combination of groups because we didn't want to administer too much drugs to the same animal because like then we could like not be like completely sure that they are like the circuit.
Yes, but so why didn't you do it between subjects then? I mean, you didn't want to administer too many drugs, so you could have done different groups of animals receiving different groups of different antipsychotic and then compared between subjects.
No, at the end...
Why didn't you do that?
At the end it's mostly that, because like most of the groups contain different animals. So we did it between different groups, but the problem of that is a methodological, like to use more than one drug on a mice is most that like surgeries take a lot, like take time, the recovery of the mice takes time, and it would be like at the end not ethical or a waste of like to do a surgery that like they have recovered for a week for just like one experiment with one drug. So we try to maximize the use of the animals. We thought that three drugs would be enough. And then the statisticals is more done, there's different groups. So now some animals have repeated, but not all of them. So
in other words, the design was like post hoc, kind of. The design was planned, but then at the end you had problems, and then you had to kind of...
in that part, like, and the baseline and PCP, like the psychotic like state, it's a completely separate group of mice. That one it's...
Right, the first one, no?
The first one is a separate
yes.
group of mice.
Yes, I was wondering because in the statistics, it's kind of difficult to understand why sometimes you compare with baseline and sometimes you compare with saline, and it kind of seems like it doesn't have a pattern, it's very ill explained in the text. Okay. And so, baseline is good because you kind of characterize the first time, but then at the afterwards, I don't see the need to use the baseline measure when you're doing, PCP. You just compare it with saline. Yeah, so why do you keep comparing with baseline all the time? I'm not, not sure.
You mean in like in both parts, no? Or like
in general, in the first, for example. Sorry, in the first, I'm talking about just acute first.
Okay.
You have a baseline measure in the first part where you compare it with PCP administration just to characterize what PCP does. But then when you do the pharmacological experiment, you have baseline again, then you have saline, right? You do saline first, but then some of the comparisons are with baseline.
Yeah.
Instead of with saline. I don't understand that.
Like you mean the saline without PCP, no? Like the first
right,
saline.
Right, just saline, saline.
We thought like because after saline and in this long protocol that it's like an hour, there may be like some changes in the locomotion, and we thought that baseline was a best description of the circuit. And then within the different groups, we compare it with the PCP saline group in the same time points. But in this sense, in the acute, also we decide, because after saline we may have some effects in like the Christmas or the mice like after 1 hour being in the cage start like get bored.
Okay, yeah, well, yeah, that is a problem, isn't it?
Yeah.
Okay, and also here in this acute experiment, PCP, acute PCP. So you have these antipsychotics, and I see that you used only 1 dose of each, which is. I wanted your idea about this because it kind of, you know, doing pharmacology and so just 1 dose is kind of tricky and very risky because you might have just not, you know, found an effect because of this. You know? So I was wondering if you could I know that you did a previous experiment with some of these drugs, some of these antipsychotics. So how did you actually choose the dose of each one?
We had like previous experiments with other, especially risperidone. I think I have it here with like a higher dose of risperidone, like 2 milligrams per kilo.
Right.
In which like the inhibitory effects were super, like way higher than the acute. These 2 milligram kilos, we thought that like, in this sense, it was like the only drug that like after trying first with 1 dose that was 2 milligram kilo, then we reduced it because we saw that there was that. And then I agree that like maybe trying other doses with the different drugs could be like, it would be good and necessary. But in these electrophysiological experiments, it also have that like you have a certain number of hours, like it has a lot of cost, and we based our like drug injections on the literature and previous experiments done in mice without PCP. We tried to base it on that. Then, like, of course, I would like to try like 10 different doses and choose do a curve dose and see which one produces the best effect. But in this case, that would be a lot of animals, a lot of mice. So we decided to focus more on what was in the literature. And we tried, in this case, we did this drug. We see that the inhibitory effects of this risperidone after PCP were really strong.
But you had it with the 1 milligram per kilogram as well, no? This disruption or with risperidone alone?
Yeah, I have like 2 milligrams and 0.5 of risperidone.
0.5, but you used here 0.5. I'm sorry, yeah, with 0.5, even though you reduced a lot, you still had these big, I mean, so my question is, you know, this is difficult because you have PCP that already disrupts the whole circuit, and then you give another drug that does something different. You know, I was wondering, you know, it's difficult for me to understand why, for example, you didn't choose to give the antipsychotics before PCP and stabilize that, or maybe a dose that didn't have any effect on neural activity, just to see if that was, you know, because I mean they're not acting on the same system, so. I don't know, what's your feeling on that? It's kind of pharmacology, but...
Yeah, no, I know. And yeah, I think like all these experiments are really interesting. In this case, we wanted to mimic what was more the reality of patients. At the end, they have the psychotic break, and then they have
you
give the antipsychotic. That was like, in our thoughts, like what was more relevant for the clinical than try to prevent it, or that would be also really interesting to see what happens if like the order is the opposite. First, you try to the risperidone and then like PCP. But in this case, we wanted to go through what it's more happening in the clinic.
Okay, yes, definitely, very good. So, and then let's go to the second protocol, the subchronic, which was the one that kind of, I would have, you know, I just know that you tried to summarize it in just one little, you know, timeline, but actually, you know, it would be nice to have the different groups that you used.
Yeah.
And in that sense, you say that you used 3 groups. In this case, you know, PCP risperidone, PCP saline. Yeah. So, but what about saline risperidone? Because if you did a 2-way ANOVA, and this is where the statistics come in, and it just doesn't fit, does it? Because you need these 4 groups. So you didn't have a group with just
no, we decided not like to do that group because we thought that would be a different, how like in the risperidone effect in the non-subchronic, like not psychotic or psychomimetic circuit. So that would be a different like question. It would be more if like risperidone would, because in this case we have a circuit that like doesn't have any memory impairment or cognitive impairment. So it would be if risperidone decrease memory or...
But it does, doesn't it, a little bit?
It could be.
But how did you do the statistics then, the two-way ANOVA? Because you do need those 4 groups, don't you, to do the two-way
like we did the two-way ANOVA comparing risperidone with this PCP saline, which is the other group that had PCP, had a memory impairment, and it's not recovered by a chronic saline treatment. So that's the kind of comparison that we wanted to do, that it's more in the cognitive deficit model, what happens when the memory is recovered by risperidone, and what happens when it's a saline it's administered and there's like no recovery of the memory. So that's what we...
So you used a t-test in this, or I mean, I just don't know.
No, well, there's these 3 groups. The first one is PCP plus risperidone.
Yeah.
The second one is PCP plus saline.
Yes.
So that's the group that I would compare. And then we have the control group, that is saline.
Yes.
And in this sense, what we compare is how...
Yes, but in order to see if PCP risperidone is doing something, right, is you need to see what risperidone is doing by itself, no?
Yeah, like that would be another approach in that.
Yeah.
That would be another approach, yeah.
Well, yeah, those are one of the little things that, that I, and also here, you have on page 79, during risperidone, subchronic risperidone, you have a plus maze, little tiny plus maze. Is that a mistake or?
No. I didn't show it, but I also did a novel object recognition task in the middle of the treatment to assess how was the recovery. But there was a partial recovery, but in this thesis, like it would add more complex, and I decided not to. I also have done a test 1 week after the risperidone treatment for the mice that could resist this long protocol, also to assess if the cognitive recovery was still 1 week after.
Okay, exactly, the carryover.
But this is a really long protocol. Right. It takes around 2-3 months.
Yeah.
Implants start to be damaged. That's one of the main problems that I had during my whole thesis, that animals don't get to the end. In
that sense, that I wanted to ask you about the final kind of protocol with the event-related potential. So you did that at the end of the subchronic treatment. So you first did the novel object, and then you did the event-related potentials in the same animals, because it's not really clearly stated in the book.
In the same
animals.
Well, not all the animals have done the auditory tests were further along in the project. So there's the first group of animals that didn't only did the novel object and the open field explorations, and then we decided to add this auditory test, and a subgroup of the animals that have gone through all the protocol did all these auditory evoked potentials. And when they finished the chronic risperidone, I tried to be as close to the finish of the end of the treatment. I tried to do everything in that animals within 48 hours.
Okay.
So all right. We can have still the effects of risperidone.
Yeah. Okay. Okay, Cristina, and what about the? I was interested a little bit in. You did the acute PCP, and you blocked it with all these nice different types of antipsychotics, but you didn't try to block the behavior with all these different. In other words, I was kind of missing that you didn't do. You only blocked them with risperidone, but you didn't have this behavioral component with the different antipsychotics, so you can associate it with the neural correlates. Yeah. So what is that? How do you view that, or do you think that clozapine and haloperidol would have Also, how would that come
Yeah,
about?
in that sense, we tried to do it with the accelerometer, but it's like not complete. The data, we only see partial or complete like reductions of the accelerometer.
Oh yeah, with clozapine?
With clozapine, it's less than risperidone, but there's a reduction of the accelerometer compared with PCP.
But haloperidol should have just...
Haloperidol, the accelerometer remained similar to PCP. So, and
the
problem is, yeah.
It didn't block the effects of PCP?
Like,
that's amazing.
Not that much. Not that.
I was kind of
the problem is
surprised by that, because I've used haloperidol and the animals are just kind of flat with that dose.
Yeah, no, I like in that case it was also surprising for us that like we don't see that much effect of haloperidol. There's an inhibition, but also as you have like seen in the video, the cable have some effect in the behavior of the mice. So it's heavy, they lose balance more than like normal mice. So it takes like it's harder for them to move because they have like all these stereotypias, behavior.
Yep.
So that like also have to be considered. And in this case with haloperidol, we didn't see that much. Well, because like I only had the cameras with risperidone, so I can like only show like these results. I have notes from the rest of the drugs, but like they are like my notes.
So you did analyze the behavior, but you didn't write it up in the
thesis.
No,
because it was like my notes, and there's like no video or like.
Right. Okay.
So, yeah, like from my notes and what I've seen, it's that like risperidone is and M100 are the ones that like produce more a sedative state, like more reduce the mobility of the mice, whereas clozapine produce a mild decrease. And then haloperidol, the PAT also produce a decrease, but not that much, like not that, and haloperidol it's also that's from my notes, but is that I couldn't like translate it into the data.
Yeah, okay, that's too bad, but anyway, thank you for sharing that. I just have two more questions, and I promise I'll leave my colleagues. I have a question about on page 189, do you have a nice, you showed it as well, a summary of everything that happened in your biomarkers and the different effects of risperidone in the subchronic treatment. And I was wondering, okay, so I just because I'm not an expert at all, I was wondering curiously, I see that in there's a lot of things that are encoded, as you say, in your jargon, with the memory acquisition. But these things don't really correspond. When an animal is acquiring this memory, it seems like there's other types of encoding that takes place after when the animal has to retrieve the information, either short term or long term. And there's only one thing that I see here. Maybe I'm not being able to read this. The directionality of prefrontal cortex hippocampus is encoded in both in memory acquisition and also in long-term memory retrieval. But it's not reversed by risperidone. So I just wanted to tell, sort of, maybe you can a little bit comment on or explain a little bit of, I think you did, but I'm not quite sure what to make of this, because is it that memory acquisition is coding something very different from long-term memory retrieval, or how does it go? I mean, just... I don't know.
Like this is, in my opinion, and this part still like needs more like trying to compare the three, like more further like understanding. In that sense, what I wanted to see is which like biomarkers were like present in the different phases. And at the end, like the first is let's look at all the things that like seems to be different between novel and familiar or early and late in the baseline conditions to describe that. And then from that, like
Yeah, but at the end, you did do all this work to code or finally record from every different little aspects of the neuron. So what does it tell you about memory? About what does this tell you? What does it tell you about memory in general, or a memory in the novel object, which is specific?
In this sense, it's more that this like functional connectivity and directionality, it's like encoding, like it help encoding or like this. It's important for this recognition memory.
Okay.
Because when it's disrupted, like it seems like this gets impaired, and
right.
In this sense, we are trying to establish which are the most relevant ones that were present in baseline and then not disrupted. But at the end, the brain also has its other mechanisms and compensatory. So what it may be doing in the baseline state, like risperidone, maybe doesn't recover it, but uses another mechanism, because we have seen that the decreased gamma on the hippocampus is present through all brain state. And this could be because of this reduction that was previously described of fast spiking interneurons. So maybe if these fast spiking interneurons in the hippocampus are dead, then the brain can no longer use that mechanism to recover memory. So risperidones act through other receptors or enhancing maybe other brain areas are also involved.
Yeah.
We cannot rule out that.
Right. And just following up, my last thing is that really in the discussion, what I missed a little bit was the fact that you didn't really associate the acute and the chronic treatments. And it's just, you had it really easy, to just kind of discuss the whole thing together and make hypotheses about what's happening when you do it acutely and then you do it chronic. And another thing is, why didn't you record at different times during the PCP so you would find, the way that, okay, for me, it's like PCP is inducing psychosis-like responses. But if you keep doing it subchronically, something changes in the brain that gives you cognitive deficits. But you didn't do it. I mean, you gave us one part and then another, and then in the middle there's just no, what do you think was happening with all that you've done and all this wonderful work? What do you think is happening with PCP? What is it doing in all these ways to produce this this deficit in cognition? And why didn't you do it in your discussion? So what is it? What's going on?
That it might be missing in my discussion. So in my opinion, PCP touches NMDA receptors, are present everywhere, are really important in the brain. And by the first dose, when you injected the PCP alone, it produced this psychotic state constantly during 10 non-consecutive days, 5 plus 2. So this produce this hypofunction that we are artificially generating by the NMDA antagonist of the NMDA receptors become chronic, and we miss when we stop this treatment, we miss the psychotic part. So these high frequency oscillations, enhancement, these strong gamma enhancement, the delta, higher connectivity in the circuit stop because the psychosis is not present. But there's some changes, in this case more that PCP produce in the circuit, this reduction of PB interneurons or dysfunction and the hyperexcitability of pyramidal cells that end up leading to this cognitive, it doesn't produce this psychosis anymore, but gets to these cognitive deficit states. And for instance, the theta gamma coupling, it's something that it's present from the beginning, day one of PCP, and then it's all present during the subchronic. So there's something happened there.
So what is different between the acute? What is it that you think is most important?
I think one of the different things is the high frequency oscillations. High frequency oscillations.
High frequency,
So the hypersynchrony at that band and the delta wide connectivity, that there seems from previous hypotheses, there seems that in schizophrenia there's a delta hyperconnectivity that comes from the thalamus and there's enhanced delta thalamocortical connectivity, whereas the rest of the cortical connectivity is impaired. So this could lead to the production of hallucinations and delusions because the brain is kind of disconnected from these sensory inputs, and it receives like random information through the thalamus. And this is more that we see it with PCP acute, and it's more in the psychotic-like states, whereas the gamma disruption in the hippocampus, the theta-gamma coupling, have been more constantly related with cognition. So these are like these changes of the desynchronization in the hippocampus that we observe in PCP. Maybe they are more related for the future cognitive part, whereas these delta and high-frequency oscillations, it's more part of the psychosis part.
Thank you very much, Cristina, and congratulations again.
Thank you, Dr. Robledo. And now we're gonna, Miguel Valencia, if you wanna take up.
Thank you, Dr. Comte, and thank you, Dr. Robledo, for killing most of my questions. So, yes, well, but anyway, first, thank you very much, Cristina, for inviting me. Thank you, Vicky, if you can hear us, for inviting me and for allowing me to have the opportunity of reviewing this massive work, I would say. Right? Is, as Dr. Robledo said, is plenty of technological effort by means of electrophysiology, then behavior, pharmacology, and then, of course, we need to deal with all this bunch of data to try to get some statistics, no? In order to explain, at the end of the day, to have some sort of model or knowledge about these two different models that classically they go in a separate way. Either we work with the acute model or we work with the subchronic. And this is nice. This effort, I think this thesis is a nice effort in trying to join them. But somehow, and a little bit, as Dr. Robledo said, you miss the opportunity of joining them, right? Because, for example, from my point of view, and after reading the thesis, right, maybe 3 years or 4 years ago, I wouldn't say the same, but you miss the opportunity, for example, of doing a recording the first day of the subchronic treatment, because you would have an acute recording for one of the mice that some weeks later you will try to characterize, right? This would be really nice. I don't know if you have some recorded from a limited number of animals, but this would have been very, very nice to do that, right? To do a follow-up, the transition between the acute and then to the subchronic, because you're using the same, and this is kind of surprising for me, you're using the same dose for the acute than that for the subchronic. And I don't know, I'm not a mouse, I work more on rats, but in rats what we used to do is, for the subchronic treatment, we use a lower dose for this 7-10 days, 2 weeks of recordings. And then, what I would have done also is to, in the middle of the subchronic treatment, to try to evoke an acute crisis, right? By means of a boost of this would replicate somehow what some of the schizophrenia patients suffer, that is kind of a crisis and psychotic effect and psychotic episode. And this would be really interesting because you have all the electrodes, all the technology, everything. And I agree with you, you want to get as much as you can from the system. So I have to say that I spent quite a bit trying to understand, to read it. There's a lot of information, and I like it very much because it's somehow the way we try to work in our lab. So I have plenty of questions.
Okay.
So I want to begin by the beginning, that is about the setup, about the electrophysiology recording setup. Okay. So you have several electrodes, 2 of them in the prefrontal, 3 of them in the hippocampus, and then you have several reference electrodes here and there in the how are the LFPs recorded? Where's the reference for the LFPs? Are they reference to this reference electrodes, as you say, or are they reference to the cerebellum electrode?
First, from the first part that you said,
sure, yeah.
I have to say that it's not the data is there because the acute injection and the baseline, the acute injection from the baseline on PCP, it's from the animals that end up in the subchronic. So I miss maybe having the comparison, and it would be a really interesting thing to do. And maybe because like at the end, I use the same mice for the psychotic-like state followed by PCP. That's why we only did baseline and PCP and not saline controls in the middle, because we wanted to use them for the subchronic. But maybe I would have to do that, like compare it with the. And I also think that it would be really interesting to maybe try to do this relapse. The only problem is like the protocol, it's already so long. Yeah. So how long should be the washout before starting for the chronic risperidone treatment? And it's that I lost a lot of animals during the way. So at the end, like it would be like try to do as much as possible in less time, so that would do. And about the electrophysiological setup, we have a ground that we reference everything in the cerebellum as a screw. We also have this other reference, and this other reference were mostly used for the single unit and multi-unit activity to extract the noise and like the neurons that were not belonging to that.
I was supposing that because I was concerned about the fact of using different references for the left piece, because then when you're trying to work with this connectivity measurements, once that you change the reference, you're changing the delays and everything, and this would have been a mess. But okay, happy to hear that. By the way, you have 2 electrodes in the prefrontal and 3 electrodes in the
3 prefrontal,
2 in the hippocampus. But you just report one signal. What did you do? Did you aberrate it? Selected the best one? How did you proceed with?
Yeah. The electrode implantation, it's an stereotrode, and one single electrode in the prefrontal cortex, and an stereotrode in the hippocampus. So at the end, what I do in all the process is I do channel by channel or combination by combination of channels, and I average them. Okay. Mm-hmm. So we had another electrode, for instance, like more back in the hippocampus. That was another, but like I haven't used because I observed that it presented like different connectivity results. So I didn't thought that like maybe it would be interesting to see also with like another part of the hippocampus connectivity changes and everything. Okay. Yeah, like at the end I did it channel by channel and then averaging all the signals.
So I was asking because you can also do a bipolar derivation in the prefrontal, in the hippocampus, giving you a more local information, but anyway, it's okay. Good. Related to the what we were discussing about the movement or not movement with the haloperidol, it was kind of surprising to me too, because in our case with rats, again, when we use the haloperidol, they stay quiet in a corner in the box, and that's it, they don't move. And maybe this is because you use the accelerometer signal in order to assess the behavioral state of your animals. And I think this is good because you don't need to do video recordings, but before using accelerometer for doing the behavioral assessment, you must be really, really sure that you are able to detect the behavioral states in an accurate way. And for example, if you go to the figure in the page 85, I think you saw it also in the presentation. The point is that when you have the animal under PCP, can you see, and you measure the distance, you see that it stays, the distance, the centimeters that the animal moves as the time goes by is constant in a high level. Whereas in the accelerometer, you see that it goes below the number. Yeah. This means that you have 2 different behavioral states. Maybe it might happen that in one case you have a translation of the animal. I mean, it is actually moving, but in the other one, it can be just heading like this, and this gives you an increase in the accelerometer signal. So maybe you should reconsider the way you are segmenting. There's a very nice paper from this guy, Rui Costa, that they are able to do segmentation, and it's not difficult to do. We've done in the lab. Once that you have the accelerometer data and you have the behavioral data, you can estimate a model in order to say, okay, this animal, according to the X, Y, and Z values of the accelerometer signal, I can say that it is quiet. it is moving, or it is quiet in a place, but moving the head. So then you have several things. Because under alloperidol, it happens that the animal is quiet, it's not moving, but it's kind of like this. So it gives you sometimes a lot of signal, right? So this could be a good idea if you want to go further. Moral methods, I would say for the cross-frequency, bunch of the methods, a lot of analysis, I like it very much, and I like the pictures, these big figures with so much information. It takes forever to understand them, but gives you a lot of information. I like them very much. For cross-frequency coupling, I would suggest you to do some sort of surrogates. You've done surrogates for the connectivity measurements by kind of shuffling the trials, but it is always very, very important in cross-frequency coupling trying to do some sort of surrogate, because sometimes you have some kind of spurious or basal surrogate cross-frequency coupling, and it's very, very clear, for example, in some of the pictures you have. You know, in some of your cross-frequency coupling, for example here, this one, it's very likely that it is something that is an artifact.
Yeah, no, it looks like.
And on the one hand, it can give you a bad idea, an incorrect idea of what do you have, and on the other hand, it can shadow very good results that you could have, right? So it's important to do that. It's very easy, I can explain you later, but it's just a matter of shuffling a little bit, moving a little bit the signals, so then you break all the cross-frequency coupling structure, right?
Okay. Yeah, no, I would ask you about that. Because
Yes, I think
to do the
now
survey.
the point is that you have a lot of data, very nice paradigms of recordings, but I think you can get more than you have in some cases. And I agree with Dr. Robledo that it's kind of a transition, right? At the beginning with the acute model, and then you have the subchronic. And it does so also in the way you treat the information. Even if you have the same information, you have the recordings, you have the behavior, and then, for example, at the very beginning, you have these big figures with cross-frequency coupling, giving you and together with connectivity, together with a power spectral analysis, and this gives you a very good idea of how the state of the brain is in that moment. But then, when we arrive to the subchronic ones, we have just box plots, very simple. Why is that? I mean, it's...
Yeah, no, I would, I've tried, to make these, nice color plots. The problem is, that it's, they are way shorter amounts of time. So the frequency resolution cannot be the same, because, we only have 1 second.
Yes, yeah, I agree for the TMAs, okay?
For the TMAs, I only have 1 second of time. So when I try to make, for instance, this plot of we need to use in order to obtain results, we need to use broader bands. This could be doing with a 2 hertz distance, but in the 1 second, you at least need, I think, for having something nicer, still need 4 or 5 hertz. So for gamma frequencies, it's could be visual.
Yes.
But for lower frequencies, all the visual part was lost, and then I struggled with that to try to make these nice plots with lower temporal. That's why with the phase amplitude coupling, we use the Tor function in the longer periods of times, and then the one on slow paper for shorter to have these temporal resolutions that allows to...
Yes, but what I mean is that you have a very nice description of what happens in the acute situation when you inject the PCP in one animal for the acute model. I would love to have this very same description for one animal for the first day after the washout, after the PCP, right? This one. What's the state of the, so to say, chronic schizophrenia state model in these mice, right? This would be, because this allows you somehow to do the link between the acute state and the chronic state. That would be nice.
Yeah, no,
you can do it, of course,
I
later.
agree,
and this is for the next paper, right?
Yeah,
so just ideas, comments, not more. And finally, about the bucket potentials, how confident are you about the results you have, about the auditory thing?
Well, there's some things that are more clear than others.
Mm-hmm.
There's like, we are recording in the prefrontal cortex and not auditory, which like may alter the results. And then there's results that I'm more confident than others, like the like the part of the auditory evoked potentials, because I have maybe a higher end of baseline and SPCP. Mm-hmm. Compared with like the sailing groups and the contours that have a lower end, I'm more confident in the part of the late components, because I that in the mismatch negativity is that because I have it in the 6,000 hertz as target, but not in the 8,000 hertz. I have like still like to process why is that, like understand why is that, what is happening, why I don't have mismatch negativity in the 8,000 hertz frequency. So I like have to look if like maybe there's some animal that it's, or maybe there's something like, you know, that like it's not.
Yes and no. I would say that you don't have an enough number of trials for the recording. We used to record for 2 hours for the mismatch negativity, and I think you did for just 20 minutes or so, and not more. So
yeah, no,
you need plenty of trials for getting a very nice mismatch response, a very clean recording. So this is something that you should consider. And the same applies for the ASSR. The ASSR you just have 50, and I think it's not enough. So maybe you would need longer trials and higher number of trials than you would have, because it's surprising that it's something like classic in patients. You have a disturbance in ASSRs, and in this nice model that it seems to reproduce many of the phenotypes, not to be able to reproduce this ASSR, I think it's a pity. And
it was like that was one of the more surprising things, like in this part of the results.
But as you said, you are recording in prefrontal cortex, you are doing a low number of animals, and indeed you are doing a low number of trials. And well, I have many other questions, but we will discuss later. So thank you very much, Cristina. Thank you, and congratulations, all of you.
Okay, thank you, Dr. Valencia. So now it's my turn. So I'm also gonna congratulate you for your thesis. I enjoyed it reading. I congratulate your advisor, Vicky Puig, who's hearing us online. So I think, again, I concur with the rest of the committee that it's a very good display of techniques that is rarely seen together. So it's very nice to be able to see electrophysiology, pharmacology, and behavior together in addressing something as important as this that usually has been looked at most separately. So that's, and that I presume it's been, a lot of work for you to learn all these various techniques and be able to, to master that for your thesis. And the thesis shows you've achieved a very good mastering of all these techniques. Having said that, the data is also, very informative. There's a lot of data, and that's maybe part of the problem, is what to do with all this data. Maybe my role is to go into that direction, since you invited me as a computational person. So I'm afraid. So I guess you got a pretty good committee selection here, where we've been touching quite incisively into the various parts of. So let me, go into my side here. But I also want to go a bit through the thesis, and in terms of introduction, I generally liked it. It's a hard introduction to make because of all these. You're going to have to cover from human clinical stuff to electrophysiology in animals to pharmacology, behavior. All that needs to be packed in. I would have liked to see more graphical support to help the reader. That's always a must in an introduction. I wrote down, that I saw a bit, you're using a lot of superlatives, which you usually try to avoid, like, I don't know what it was. I wrote it at some points, I was like, no, that you should try to avoid. Some causal relations with schizophrenia, like this a band causes that. Yeah. I think that we have to stay away from for the first time being until we see your data, I guess. I also found some confusion with the word model, because at some point you talk about model, computational model, but your thesis is really about model, animal model. This often happens, that then so that happened in the thesis. One comment on the hypotheses. I found the hypothesis in page 48. There's one that I figured that, let me see. So on page 48, the first hypothesis is psychotic-like states induced by acute PCP produce major alterations on prefrontal hippocampal neural activity and synchrony. I wonder if the causality is inverted here. Well, is your hypothesis that the neural activity is a result of the alteration of the psychotic state, or is the other way around?
Yeah, no, that's
I guess. Yeah, sometimes I thought your real hypothesis is the other way around. That would be the fun.
Yeah, the major.
I
agree that it could be also inverse.
Okay. So. In terms of methods, so I'm not an experimentalist with animals. So I guess the rest of the community understands it much better. But I see that the number of animals keeps going down, or there are some tests in which there are very, very few animals. Maybe you can explain. You know, how does that happen, and why does it affect some of the experiments, not others? And you know, I guess it has to do with making experiments and how life is. But maybe you can just comment on that, because it's important eventually to draw conclusions.
Yeah, now in this sense, for instance, in the first part, the acute and everything, there were experiments that were done with a week of separation. So within 1 month, you could do like 3-4 experiments, but like the other part, the subchronic treatment, it's we do an implantation on the first day, and there's like it could take between 2 or 3 months to finish the protocol, and at the end, like implants lose stability over time, animals hit, like with the head hitting, so implants fall. Also, there's a lot of injections, which it's a risk for the animal too. So it's inevitable that the longer you make the protocol, like more animals you will lose. And especially it's that like in the subchronic part, we have baseline characterization at the beginning that could like take 2 weeks, 3 weeks. Then we have a 10-day treatment that takes like 2 weeks more. Chronic PCP characterization again, and then a chronic risperidone, and all of that causes like this stress on the mice that like we could lose them, or in the implant too, which like may damage like the signal. Like I lost, like even the implant remained stable, the signal could not be compared with the baseline because there's gliosis, there's like inflammation, could be some of that changes like over time. And we may, especially the single unit and everything at the end of this protocol was like really hard.
So in that sense, I found like when there were these few animals, it was important to actually show all of them. And you do in a few, in many of the graphs, but some don't have that. And then, you know, an average of 4, you wonder what is the variance you see. So I think that is something that would be useful to just be very, very transparent there. Yeah, that's the data you have, and if it's actually very consistent across animals, I guess it's quite different than if it's just the mean, but one goes this, the other goes that. So that's a part of the transparency that would be nice to see. It's in many graphs, but some of them I didn't find.
I tried like in the box plot, like that's another thing why I use box plot, because I can there's the dots and the lines.
Yeah.
So it could be more representative. If there's 4 animals, you also only see four lines, and it's more the consistency that the 4 lines go in the same direction.
Okay, then here, in this graph here, what is it? This is a single animal, or are we looking at averages here?
This is average.
This is average across animals.
Yeah.
Also the spectrogram.
Yeah.
So you average spectrograms?
Yeah, we average at the end, we average spectrograms, yeah.
You average spectrograms?
Yeah, that's only for visualization purposes, for statistical analysis.
Okay.
We did quantification.
And then the error bars that you put, that's across animals.
Yeah.
Okay, so you get really small variance across animals in your power spectrum.
Yeah, no, especially in this part, in the PCP acute results, the effects, like the pharmacological effects, were so strong that it's really consistent across animals. Compared with then if we go to the behavioral results, there's a lot of variability, and then we got to you go to the other part here.
It's fine.
Yeah, the bars are
yeah.
Here there's a lower number of animals, and the bars were way higher.
Okay. So back to the one where you were at before. So maybe you can help me go through those spectrograms. Somehow you look at those spectrograms. No, not this one. Yeah, well,
this one is the most here.
And you tell me that risperidone recovers, but I somehow I don't read that. So I see the spectrogram before, either in baseline or after saline, and it doesn't look the same as after risperidone. So how do you interpret that?
It's not the word probably it's not recover. Yeah, the word it's not recover. It's suppressed what we think it would be the psychotic biomarkers. These enhanced high frequency oscillations, they go down in frequency and down in power, and then gamma. The problem is this is also zeta score spectrograms. So this is changes, and this is one-fold changes. So at the end, you could in some of the spectrograms, there's maybe 7-fold changes, and it reduces it to 3 or to 2. So there's a reduction, but maybe in try to combine all the bands.
You do the spectrum.
Yeah, it's more.
I wonder if you would plot on a logarithmic scale the power, which is the common practice, if that would
we
help
also have
in visualization.
The problem is that high frequency, in that, we discussed a lot about how it could be visualized and what is the best way of plotting it. And when you plot the power as raw power, as I have it here in the beginning, the problem is when you get to high frequency oscillations, which is one of the main changes, due to they are faster oscillations, they are less visible, the changes, so you miss that part. So we will trying to be able to see.
Yeah, it's hard to find the right representation to convey.
In the first PCP one, here, and this one. So here, in this case, but here in the hippocampus, you can not actually see this decrease, that it's more clear in the zeta score than.
Oh, this is already in decibels, no?
Yeah, this is how it would be seen in the raw without zeta score. This is how it could be related to the zeta score, and is that you in this visualization, especially what happens at high frequencies or here.
Left is raw and right is this score.
Yeah.
Okay.
So you could is that at the end we choose this way of more to see the changes.
Yeah, Then I have a question of how do you measure behavior in the memory task? So if I understood correctly from your methods, you're watching the animal, and with the joystick you mark whenever the animal either looks to it or approaches. Or no, or it has to be within a certain distance?
Yeah, it has to be, like, we try to follow, as described in like the paper, I think from 2003, that it's like the rules for like the novel exploration, that the mice had to be like pointing the nose, like at a certain distance, no longer than 1-2 centimeters, and they can, like, if they climb it, you stop like pressing because they are not like looking. It's something like you can try to automatize it, and there's like now the Black Cat and all these softwares that tries, but like need learning, because it's not as like the mice have like to be this close to the object, because sometimes you could like these softwares have like, because the mice is like sitting in front of the object but not exploring it, and the software would count it as exploration, and the mice is like at the end it's like had these mistakes that like
what I noticed that you said that in some cases it touched the object, in others it didn't. I'm wondering how, I guess that's nice, that's both exploration. But when you were going to look at neural data, I guess it could be totally different to touch or not to touch. And yet you don't seem to consider that specificity when you do your neural data analysis and compare. What if, it tends to touch more at the beginning than at the end of the session? Or what if it tends to touch more the familiar than the novel? And that's what that was a sort of concern I had when I saw that you were putting together things that involve different sensory conditions.
Yeah, no, that's a really good point. This sense, like, I feel like it would be a really interesting thing to look at this difference, but like, as that like would involve like a joystick with more buttons, like more human failure. Unless you automatize it, it's like, okay, this button is right, but not touching. This button is like right touching. This like it would, and as long if it's not that automatized, you could like do it with the video, watching like.
Yeah.
You could like spend like, or automatize it, the Black Cat that I said like until now has all these problems, or you spend like a lot of hours trying to watch, like, I have all the videos, so trying to watch like a lot of videos and trying to mark it, but at the end also that adds the human error of like the buttons. So we tried, like, in this sense, maybe it's not completely the same state, but we are also not in like recording in sensory areas. We are more downstream process, like the hippocampus and the prefrontal cortex. So we thought that was a better approximation in this case.
Okay. And how do you control for possible biases of the experimenter who doing this thing? Are you blind to what is the novel and the familiar object when you do that?
This is like, that could be kind of an error, because it's really hard unless there's two experimenters, and it's not good for this kind of test that, like, because you per se as a human have some error, and if it's across across different people, the one could press a little bit earlier than the other, like the reflexes may be different, so it needs to be at least that part of consistency. In that sense, I tried like to, it's tried not to have bias. I was like putting music or putting like trying not to focus on which mice, if this was saline or PCP. I had done like many animals in one day, so at that point I was like, okay, another test. Like, I don't know if this is saline, PCP. Just like put the animal, novel object, and try like to keep like just mechanically, try to keep it video, hand, and like be my ears, and like not focus on the experiment itself. Just like that's the way I could do it.
Okay. No, I guess it's probably, That's it's just the resources, I guess. Okay, so let me, yeah, about, I was satisfied when I saw the shuffle that you do those shuffles. I thought that's very good practice. I guess you already subtracted it from here because I don't see plotted anything about that. It's subtracted, right?
Yeah, it's subtracted, and they were mostly done in the more behavioral part because like at the end shuffle like takes a lot of computation and time. So in these like acute experiments, like it was like months of like how I had coded of like shuffling.
Months of shuffling?
In this like chronic like taking account because like it was like we had a point in each minute for like 3 hours. So that like that's why. But in this case, the effects were so pharmacologically robust that we decided not. But then the behavior, because there was like a lot of more variability, we did all the shuffling corrector, and yeah, like I already subtracted, and I did the statistically before subtracting and then subtracting to like check that the results that we were seeing were.
So I agree with Dr. Valencia that this should be that. You know, extending it to the rest of the analysis would be a lot of, because it's something we never know where the small biases might be and artifacts, and that takes care of it for free, essentially, only computational power.
Yeah.
But these days, that's pretty cheap. Okay. One thing in the results. So one thing I found. And I saw in the second chapter of your results, which is chapter 5, yeah, I think. You say you already take care of the situation. You consider the fact that you wanna compare things when the locomotion conditions are about the same, because you say it's a fair comparison. You don't want to get all the motor aspects of behavior to contaminate your neural biomarkers. But you don't do that in the first chapter. And there's very strong effects of both the PCP and so what do you say about that?
Yeah, no, like at the end I took into account locomotion as like also in the first chapter. But in this case, it was more locomotion influenced by drugs that had, for instance, Respirator had this sedative effect. So it's like not similar that much to quiet as another brain state, or this hyper locomotion produced by PCP. Like I found like in the first part, I found like really difficult to compare like this locomotion, no locomotion parts with the brain state produced by drugs. Of course I took like the influence, and I checked the effects of the risperidone and clozapine alone, not with PCP, and also alloperidol. It's done in a previous paper of the lab to understand this more sedative and I think like it could be some comparison of some of the biomarkers like the reduced theta in the hippocampus.
Right.
Like in
so you could take, say, in baseline and take periods of slower and faster motion, and then see if theta goes down. Well, theta will go down. No?
Yeah, no, that's like that's.
So.
Yeah, no, at the end, the figure it's maybe introduced later, but like we also like this figure was like the locomotion one that it's. I also have it in the extras. It was more like we take it into account, especially this theta in the hippocampus, possibly the theta gamma coupling. But at the end, PCP produce hyperlocomotion, and theta is completely disrupted, and theta gamma coupling is disrupted.
Yeah,
so
it goes the other way around.
It goes the other way around. So there's like such strong pharmacological effects that I think go beyond locomotion in this first part.
Okay. Yeah, it's still a confound for because you see so many things that some of them might be just that the guy is moving faster, right?
Yeah.
So I don't know how one could control and about that, especially in the acute case. In the other case, you do
It's this one.
More controls, yeah, this one.
Right here.
Yeah. Then when you do the memory, the quantification of memory impairments in PCP, I'm thinking about working memory, you find an effect. But then you look at different aspects, how many times it looked, how long, and then there was one figure that was intriguing to me that showed how the animal, so how long the animal explores each object as a function of the sequential visits. And that, yeah, here.
This is for the working memory.
Right. And then it appears like right at the first, when the animal gets on the maze and starts, it does know which one is familiar and which one is not. So it's not really an impairment in memory, what we're seeing in these animals. It's just that they lose interest very quickly. So after the second or third visits, they're already not caring which one was more. But they know, it's like they're less engaged in the task. But when they get in, so this goes also into something that I found somewhat, I don't know how you interpret that, but you mentioned working memory, and you're testing 3 minutes after there's acquisition of the memory. For me, that is retrieval. It's you are getting the information and doing something with it, rather than keeping the information. So the first test is really checking whether information is there or not. From then on, it's all about exploration, interest, or how much of this is working memory, or is a defect in working memory, or a defect in retrieval or sustained attention, sustained interest.
Yeah, no, that's a really good point, because I also thought, because you see in PCP you see that in the working memory, but like then in the retrieval results, it's more like in the long-term memory, it's more
clear here.
More clear that they don't like know which one is it. And I kind of like try correcting with that, like just choosing like 5 seconds of each, just like to discard this effect of like, okay, these are the first explorations of the mice to the objects. They are retrieving the information, and not like longer exploration that would be more affected by the interest or the attention to the objects, because as it's clearly seen, mice decrease the duration of explorations over time, probably because they lose the interest. So in that sense, it could be, because I was also surprised about this, that in the PCP, it seems like these first 2 interactions are, and then it like decreases like super fast. And it's a good point.
So it is an impairment. There's something different.
But
I don't know if I would call that a problem in maintaining information over 3 minutes. It seems like the information got there, but then something else about that.
Disrupting this.
Yeah, which is the memory for 24 hours does seem disrupted. There, the first exploration, they don't distinguish familiar from novel, but here they do. So I don't know. I don't know. Yeah.
Yeah, no,
also in that direction. So I was, when I was reading your behavior, and I was thinking about the working memory and long-term memory, which I would say retrieval. You are more specific about retrieval when you talk about long-term memory. You say retrieval, but for working memory, you keep saying working memory, which I would say also working memory retrieval. And in this sense, if I would have made an hypothesis for what you would have gotten, I would have said you would get the same neural markers for the two. They're both retrieval. The animal is just getting it from different sources, but at that point which you're analyzing, it's doing the same thing. It's just using a memory to do to move and do stuff. But yet you don't see it the same, which is interesting. And you see something that makes sense, this prefrontal here and the two directions. So I think that's an interesting result that maybe could.
Yeah, no, it's like.
So when I read it, it's going to be the same. It should, that would have been my prediction. And instead, you found similar things, but you found a difference that makes sense too, like prefrontal versus. So I think that's neat. Okay, so I have many different questions here. So going more broad now. So as I started saying, my sense after reading this is, this is overwhelming. There's a lot of information. There are all these tables of that Dr. Robledo was going through, all the crosses and it's really hard to get a full picture of what is it that is going on and what is important, what is not. And I would like to see if you can make the exercise to to organize this a bit for us. And I would give you some possible ways of organizing this. So one way would be, you showed, that some of the results come from a larger sample, others from a smaller sample. Some you might see and you have a sense are more consistent across animals, others less. Others, in some, the saline didn't quite go well with the other, and there was which would, so based on all these things, some of those crosses maybe are, they're more of, these are hypotheses for future experiments rather than something that I'm going to put on the table like this is solid. So are you in a position to say which ones of those crosses and tick marks would be based on, this, there are more animals, they're consistent across animals, and the saline is the saline control goes in the direction one expects. What would it be that you would say, acute?
In that sense, it took me a long time because going from because there was many things that were changing, then we reduced to the ones that also were impaired by PCP to understand if it's impaired, it's not working, probably it's because it's relevant, and then what also correlates with the memory performance. So in this sense, I see more consistency in the direction, the functional and the directionality results at these theta and gamma oscillations. And the most consistent thing is the disruption on the hippocampal gamma and theta gamma coupling produced by subchronic PCP. That it's present always in all brain states. It's not in saline. And it's not, and then it's not recovered by risperidone.
Right, but that's the problem, no? What I noted here is, after seeing this, I would say, the hippocampus has dramatic effects, but they don't seem to go with behavior because risperidone recovers things, and yet this particular biomarker is there. Is there on. So it doesn't seem, it's something that it's clearly an effect of your drugs, but it's probably not related to all the measures that you make that we care about in terms of behavior. And that's the nice thing of your thesis, that you can link. So wouldn't the result be that prefrontal is where
all the...
It seems like your prefrontal and your connectivities, which you relate that more to behavior, whereas this big effect is there, but...
in that sense, for the behavioral results, I think it's more in the directionality and how this prefrontal and hippocampal directionality and all of that, it's more relatable. The thing is because risperidone, it's a drug that you administer globally and it may affect other like circuits besides the prefrontal cortex. It may be activating other compensatory mechanisms that are not present in baseline because there's this massive disruption, and it could like do it through other circuits, there would be like another effects. So I would like in this sense to try other drugs that are more procognitive to see, because risperidone at the end it's an antipsychotic that the main effect is to stop psychosis, and then it has some procognitive part, but it's not the most procognitive antipsychotic, like not by far. So it has like also a dopaminergic component, the two, that it's been proved that that's not cognitive. So I feel like in order to be able like to understand more how this cognition is resolved, we should try. We are currently trying more procognitive type of drugs that like maybe rescue more like the baseline circuit instead of like touching like everything and activating the brain, like maybe producing another activation. So we are already touching other serotonin receptors, the serotonin 7 or the serotonin 4, using another antipsychotic drug like lurasidone that it's more procognitive, in order to understand that maybe is that in the terms of like the cognitive recovery, risperidone it's maybe not the best drug, an example, and still it can recover this aspect in these mice, but like it probably doesn't recover other things.
I see. So you say the risperidone effect, you wouldn't take it as the main. I see. So let me put it in a different way. It's a similar question, but suppose you're now, you take up a postdoc position somewhere, and you love your data. You take up your thesis with you, and Vicky is happy with that. And you team up with a computational postdoc, and you're going to try to test some of your discussion about how things emerge from, disruption in inhibitory neurons and excitatory neurons and connections. And okay, and now you have to decide with this other person, where are we going to do a single area? Is it going to be prefrontal, or is it going to be? Do we need two areas? And what is it that we're going to? What would be the one thing that you think it would be? Is it this theta directional connectivity? Is that the high gamma prefrontal? What would the one thing that you think there's better evidence from the molecular cellular side that would link to something that you think is interesting that you have found and that you have data for to see how a circuit actually eventually reproduces this?
Oh,
that's a hard one.
Yeah, no, it's a hard but a really good question. And in that sense, is that I would think that like the connectivity between both areas it's important for behavior, so I would like go through that direction. But at the end, these like directionality are it's a biomarker. So it's like a general, and I think like we are like currently also doing it. We have all the spiking activity, and we want to understand how these like cells can be associated with these gamma defects and everything, and how like these both areas are like more a spiking rate, and understanding like why like more mechanistic in that sense. And if we had like the data from the cells, like we can more understand. I feel like we need that part to finally understand.
So you think it's too early for a computational model?
You could do some try like once like with these biomarkers, and especially the ones I feel like the functional connectivity ones, the prefrontal to hippocampal directionality, to assess like how they could like predict or like can like the memory performance, seeing that correlate, especially the ones that correlate with the discrimination in that are correlated with more. Try to understand how this could like mechanistically work in this sense, and how could be disrupted by PCP. I would take into account this baseline and PCP condition, and for now, leave aside the risperidone part, which it's another level of complexity.
Okay, good. Okay, I think I'm done. I have a few more questions, but I think I'm quite satisfied with your responses for now. So if there's any question from the room or the chat on the YouTube channel, now's the time. No question from the chat. Some question in the room for Cristina? No? Okay, then I would ask the audience to leave the room and let us deliberate while we reach a consensus, and then we're going to call you back in.
Yeah.
Thank you. So this committee has deliberated and has unanimously decided to give you the grade of excellent. So congratulations.
Thank you.
Okay, so now she doesn't need to sign anything.
Yeah.
No?
She does?
No.
I don't know.
No.
No signatures.
Not sure.
Okay. So then I think you'll be all set and you're gonna...
Yeah, there's a Pika Pika in the third floor, so you are all invited now and you can follow.
So congratulations, Dr.
Congratulations.
Delgado Sallent. Is it Delgado Sallent or Delgado?
Delgado Sallent.
Delgado Sallent.
Delgado Sallent.
Congratulations.
Thank you. Sí.
Ya está.